But welcome to the afternoon. So we've looked at different aspects as we've gone about how we can diagnose better, how we can come up with better treatments. Perhaps now we could start and take a panel and look at how we stop getting ill and look at prevention so we don't need them. Not sure we'll be 100% successful in that, so we still will need other treatments, but actually understanding how we stay healthy, which came up in the previous panel session, and then looking at the session that's entitled The Good, the Bad and the Ugly. So I'm delighted to hand over to, I'm going to say Clint Eastwood, but it's not actually, it's Richard Vickers, who's going to take us through the panel for the transcript.
Well, hello everyone, good afternoon, welcome back after lunch. Welcome to the fabulously named Good, Bad and the Ugly. I wish I could take credit for that name, but I can't. But I think it's a very pertinent area for us to be talking about, especially listening this morning to areas about AI, diagnostics, health, etc. It really does overlap and resonate with many of the things I hope the panel will talk about today. And infection, I mean, come on, we've lived through COVID for the last 2.5 years. We know the huge impact on human life, impact on the wider healthcare system, economy, social structure, political. It's a huge impact for us. But remember, COVID-19 was only the third novel coronavirus of significant clinical concern to be identified in recent years. Look at the issues we're having with Ebola in West Africa, things like Zika virus coming out of South America. We have this huge problem and burden of infectious diseases throughout the world. And when you start to look at developed countries, particularly my area, and the thing I'm really interested in is resistance in nosocomial pathogens, especially bacteria. And we are now seeing bacteria and infections that are pan-resistant. We are having to resort to medicines that we haven't used since the 50s or 60s because they're simply too toxic. And we are having to use these now. And there's a huge unmet medical need. If you read the O'Neill report, it's estimated by 2050, if we don't address this, there'll be 10 million deaths a year around the world from drug-resistant bacteria. And I don't think that even includes things like TB, malaria, and the issues we have in the third world. So we have a whole group of very, very exciting scientists and areas to address, but there has been a difficult commercial model, undoubtedly, in infection, despite everything I've said, despite the efforts of all the people within the community. And I think there's lots of things we need to do to address that. And what I want to hopefully get through today is maybe some reflections on the past, a kind of where are we today, and then the very dangerous topic of crystal ball gazing, where we can start to think about what are the areas we can push into. We've got a very, very diverse panel here coming from very, very different backgrounds. So I'll shut up now and I'll hand over to the panelists. I'd like you guys to give an introduction to yourself and who you do, what you do day to day, what fills your inbox, but also maybe pull out a couple of topics within the field of infectious diseases, not just AMR, but just broadly speaking. And then maybe we can start to examine and probe some of that, and we'll take it from there. And perhaps if I go to the subs bench and go to Paul first.
Yeah, thanks, Phil. I am the substitute for Michel de Baar, so at a very late stage. You got a bad back, I got a bad back, I'm still on the panel. So background, I'm a pharmacologist, been in the industry since 1979, which dates me somewhat. I have been working in infectious disease for much of that. I was part of the, so if anybody in the know, part of the metronidazole group back in the day in anaerobic sepsis and areas like that. From the sublime to the ridiculous, I was also part of the Lamisil development team. For any of those of you who've got athlete's foot, that's the one to have. But importantly, within tranScrip, this is almost our DNA. We've worked on 7 of the last 8 major antibiotic developments, so this is our space, absolutely. And that's my wife just pinging me a message saying good luck. So I'm going to address the sort of business elements of the failure to make a commercial case for infectious disease treatment. But I'll also listen in and give my penneth on anything else that I feel I can contribute to, bearing in mind I'm a substitute. So.
Thank you, Paul. Maybe Mike.
Wonderful. Thank you. I'm Mike Whelan. I'm a project leader at CEPI, the Coalition for Epidemic Preparedness Innovations. You may or may not have heard of us, but we were set up in the wake of Ebola to make sure there was going to be no major pandemic. Well, that worked out well, didn't it? But actually, there was method in our madness. I'm actually an immunologist and a biochemist by training. I've been in the vaccine industry for longer than I care to remember. But when we set CEPI up, as I say, it was really to learn the lessons of Ebola and to be better prepared. And to a certain extent, we were. You both mentioned the business case. We work on diseases with epidemic and pandemic potential, which are largely ignored. Just because you're poor and live in a low to medium income country does not mean you should die. So we have to do something about it. And also these viruses have a very strong propensity to mutate. So we started with quite a defined portfolio of Lassa, Nipah, chikungunya, Rift Valley fever, and MERS. And I deliberately put MERS at the end because it was a beta coronavirus. And a lot of the work we did on MERS, we were able to translate. We were actually working with Sarah Gilbert on MERS way back then. So we were able to move very fast. We also prepared a lot of platforms and so forth. So we were ready. And when the pandemic struck, and I have to take issue with the word that was used this morning, the pandemic was not fantastic. It was used this morning. It was absolutely not. There were over 1 million deaths, and it caused trillions and trillions of dollars of damage. We had the biggest portfolio of COVID vaccines in the world. We had something like 13 or 14 active projects. We also were instrumental in setting up the COVAX network with our partners at the WHO, UNICEF, and Gavi. And the idea was to make sure that the poor did not get left behind. And let's face it, they did. Vaccine nationalism is wrong. So we set up COVAX to ensure that. Personally, I worked with our friends at AstraZeneca, and we managed to get 320 million doses into COVAX. Of a total, by the end of last year, we got 2 billion doses for those that couldn't afford to pay. That's got to be a good thing. So it's a model. Anyway, enough lecturing of you. What I was going to say is the other thing I was going to talk about today is we've sort of gone back to the other diseases. And I've taken over 2 portfolios, one for Nipah and one for Rift Valley fever. And I think, although we focus primarily on viral diseases because they're more likely to be pandemic in nature, a bacterial probably isn't, but that's not to say that AMR is not a huge problem. It absolutely is. But what we're hoping is a lot of the lessons that we're working on, particularly with Rift, could be translated and could well be helpful as we move forward. But perhaps we could come back to that later.
Yes, definitely.
Sorry, lecture over.
Thank you, Professor. Maybe Anne?
Sure. So hi, I'm Anne Neville. I lead the discovery and microbiology team at Microbiotica, which is a microbiome company spun out of the Sanger Institute back in 2016, though we are now based at Chesterford Research Park. We are interested in developing a new class of medicines that we're calling live bacterial therapeutics. They are made of consortia of pure strains of bacteria that we typically isolate from humans. And at the moment, we're working on 2 products that are moving towards clinical trials next year, one for immuno-oncology indications and the second for ulcerative colitis. And the reason that we're really interested in AMR is because, of course, all of the bacteria that we screen to put into our medicines need to be AMR-free, ideally, and certainly shouldn't have any AMR genes and any mobilizable elements for fear that they would transmit it to another member of the microbiome, especially a pathogen. Other people working in the microbiome field maybe take a different approach to diagnostics and therapeutics. So some people, for example, will take whole stool and transplant it into a patient with a particular disease. This is very common for C. difficile and it works really very well. But the problem of AMR arises if the stool is not properly screened for the absence of antimicrobial resistance genes and pathogens, you can transfer, unfortunately, an infection to the recipient. And there was quite a famous case a number of years ago where, unfortunately, people died from having received contaminated FMT. And this caused everybody to stop and say, okay, is this safe? Are we doing the right thing? Do we know what we're screening for? Do we know how to screen for it properly? So whilst AMR is maybe not the main thing that we're working on, we certainly have a real awareness of it, and it makes a real impact in the way that we go about developing the medicines that we're developing.
Yeah, absolutely. Thank you very much. Microbiome is a real pet subject of mine, and it might not be pure ID, but if you imagine a Venn diagram, there's a very big overlap. And in fact, actually, when we get into the crystal ball section, I'd like to examine that a little bit more because I think it's a real frontier in this area. Okay, and finally then, but not last, Martin.
Hi, I'm Martin Stocks. I am an immunologist by training as well. I have spent the last 20 years working largely on the sort of academic commercial interface, discovering new technologies, taking them into commercial settings, building businesses around them. I seem almost by accident to have focused in on infectious disease for much of my career, and I've worked on projects across the board from diagnostics, vaccines, full-on antibiotics and alternatives to antibiotics, including like bacterial therapeutics, phage therapies and more. I am currently CEO of Iceni Glyoscience, a company that is a group of glyco-engineers. And we are, again, in the infectious disease space, we are engineering glycan molecules as potential ligands for diagnostics, as vaccine antigens, and as targeting molecules for direct-acting therapeutics and prophylactic molecules. I think my particular interest today, or a couple of things that I want to highlight, one, following on from the previous panel, is the area of diagnostics. There was a Science and Technology Select Committee meeting about 3 or 4 weeks ago on AMR, and they had Lord O'Neill as one of the people giving evidence, and one of the questions he was asked was, where has your report failed? What has not been done? And the one that he picked out was diagnostics. He said there has been no advance in diagnostics in infectious disease since that report came out. Perhaps a pejorative way of putting it, but yeah, I can see how that is working. The other area that I think is really critical at the moment is filling the pipeline. So this very week, the WHO report on antibiotics development came out, and the number of new agents in clinical trials, and we're talking antibiotics and alternatives to antibiotics, is now down to 27. It was 34 last year. It's going down, not going up. So there is a real problem, not only with developing antibiotics, and there's a whole piece about market failure and new models being needed to drive development, but we also need innovation. We need new molecules coming into the pipeline, and that's a major issue at the moment.
That's brilliant, thank you, Martin. And again, I think you touched on a couple of subjects. You brought up phage, for example. Diagnostics, that's something I'm really quite interested in. And I think as we get to, novel classes of compounds, not new beta-lactams, new penicillins that we've been generating for donkey's years. And I think as we get to the crystal ball stage, I think that's going to be really good to see where's the innovation, what are the innovation gaps, and what do we need to do to really get this field back onto its feet. But perhaps if we go back and we're thinking about maybe where we are today and what the last 5-10 years have been, all of you have brought up the sort of, shall we say, the market failure. I think it's also very interesting what you were saying about vaccines. I mean, I started my career in tuberculosis 25 years ago. I still think it's a tragedy, the number of people that die from tuberculosis, which we should be able to cure. And yet so we've got LMICs, where the cost is important. It's the same applies in Western countries. We demand antibiotics to cost nothing. We'll pay $50,000, $60,000, $100,000 to treat a cancer patient, but we won't pay more than $200 for an antibiotic, and they die of a multi-drug resistant infection. How does that work out? So I wonder if we could open up the panel here and we could just sort of throw some of those ideas around and think about what we need to do from that perspective, and then we can start to think about the future. Do you want to start?
Yeah. I was just, when you were talking through about the various modalities of treatment, and we've always looked at how can we kill bugs. And the thing about the microbiome is that you don't necessarily have to kill bugs, you just have to change the environment in which they're in. And one of the key elements of the bacterial formation is they form these biofilms. And there's starting to be ideas about how do you disrupt that as a way of actually managing the condition rather than trying to necessarily kill every single organism in the infection. Beyond that, you're looking at the business case. It is, and just to reiterate what you said, 200 quid for an antibiotic. The problem with an antibiotic is that it actually works or it doesn't. And we only take it for 7 to 10 days. In oncology product, you can go back and do it again multiple times usually, but you can't do that with an antibiotic. And the thing about an antibiotic is that it actually works or it doesn't. If it doesn't, the patient either dies or you go into another antibiotic, but the cost you of actually developing that antibiotic is the same as if you're developing an anti-oncology agent. and the model doesn't work. That's why there's no investment. And you're going to talk about pull rather than push.
Well, I think it's probably worth flagging that there have been some successes following on from the O'Neill report. And one of the things that I think has worked or is starting to show promise, I think more accurately, is the development of different models for reimbursement for these drugs. So Paul's absolutely right. There is no economic case with the standard drug development model for developing antibiotics, and that's why pharma has largely exited from it. But things like the subscription model that the UK has introduced as a pilot study has the potential to do that. So essentially the National Health Service will pay a company for its drug for a license to use the drug. So they pay it several £1 million a year for a license to use the drug. And then it doesn't matter if that drug sits on the shelf and is never used or is only used once or twice. The company gets the value, the public gets the value as well, because you don't want to be taking new drugs and immediately blanket bombing the whole population with them, because all that does is drive further resistance, and all of a sudden your new drug is no longer effective. So new models have been effective and hopefully will get embedded in health systems around the world and will draw sort of new development through. But I think one thing to flag there as well is that market failure is not just in the case of new drugs, it's also in the diagnostic sector as well. There is a strong argument, if you talk to sort of clinicians, hospitals, they don't see the value of doing post-hoc diagnostics for infectious disease. You have a patient turns up in the clinic, the doctor suspects it's an infectious disease, they just start them on antibiotics straight away. That's full on, bang. And what happens then is they keep them on antibiotics until, Paul says, it either works or it doesn't. If it doesn't and they're still alive, you start them on another antibiotic. What if you have a diagnostic that can say, well, actually, this is X bacteria, it is resistant to Y drug, therefore you need to use drug Z. You can get that result in 24 hours. You can stop the use of an antibiotic that is doing no good, but is actually contributing to global resistance, and you can effectively treat that patient. Or if it's a virus, you can stop that antibiotics treatment straight away. So there's real value, but there's that disconnect between where the actual value lies and where the cost comes from. And until those two things are connected, we still have a major problem.
And I think that overlaps with a couple of comments that came out from this morning's session. We need to link up the various disciplines. We need the medical microbiologists, we need the clinicians, we need the diagnostics, we need the drug discovery, drug development people to pull together to, if we could, to be able to do a point-of-care rapid test that says it's a multi-drug resistant Pseudomonas, this is my very Pseudomonas specific drug, I use it, I'm not using the sort of broad spectrum carpet bombing, and those are the innovations we need to move towards.
But we know how to do this, right? Like in theory, we could put all the different parts together. It's just, I think, sometimes barriers to making it happen and operationalizing it and training people so that they're actually going to see the need and do something about it, in addition to funding it and everything else that's required. But I mean, if you're to reflect on what happened during COVID, when everybody put their mind to something, look at what the whole sector achieved, right? We went from brand new disease, brand new infection, through to multiple vaccines in rapid time. So if you take away all of the barriers and decide you're really going to do something, I think it can happen, and can happen quite quickly, because we know what to do. We've got smart people who can drive it forward.
But the problem is that the driver for COVID was an existential threat.
Sure, but this is too, right?
Yeah, but people don't see that yet.
Well, then it's,
At the cold face, and that is the crux of the matter, is that until somebody, I say this so many times, until somebody famous dies, or a particular locus, as happened, as Martin was alluding to, in New Delhi, where you have these multiple multi-resistant forms, which pretty much we can't do anything about, but they still localized in Delhi. If they go global, then that everybody says, well, why aren't we doing something? Well, we told you. So
Yeah.
Could I come in here?
Yeah, surely.
This is precisely why we were set up in the first place. I mean, let's face it, pretty much all of the diseases we're talking about are zoonotic.
Yes.
They've come from animals.
Yeah.
We knew they were there. We just didn't look. But there is a market failure there, and I doubt the market will ever be there. In low to medium income countries, they'll never be able to afford it. That's why we need the likes of Gavi. That's why we need the likes of Kovacs. But the point is, these things can mutate, as we saw. And then we get down to this cost of goods argument. Now, I was interested in what you had to say, Martin. We've been looking at this a lot recently. And yes, cost of goods needs to be very low. The AstraZeneca vaccine was incredibly low. They did a fantastic job. Moderna and Pfizer were a lot more. But the difference is, COVID didn't discriminate between high and medium income countries. It was all the same for everybody. The diseases we're talking about are essentially low income countries. Nobody cares. But if it mutates, you will care. So the question is, how do we get better preparedness? And then how do we get there? And then that's why personally, I've been very, very interested in One Health. And what can we do with that? Because it touches on pretty much everything you guys have said. It touches on diagnostics. It touches on identifying outbreaks in epidemiology. It talks about getting new antivirals, antimicrobials. It talks about vaccines. All of them are related. So perhaps.
No, I think those are really pertinent points, and it is, we need a collective around this, whether it's all the way from epidemiology to molecular microbiologists to whatever. But if we can pull it together, I think there's a way forward. You mentioned that there's resistance in areas like India and sort of low to middle income countries. Actually, if you were to look at some of the latest resistance data in Eastern Europe, in Greece, in Turkey,
Yes,
it's alarming.
Mm.
It's alarming. I mean, 50% of Klebsiella isolates are pretty much pan-resistant, so it is knocking on our door. That existential threat is knocking on our door.
We get these bizarre loci of infection. I mean, for instance, around New Orleans, syphilis is a big thing.
Yeah, that's returned to it.
Yeah, and we thought syphilis was a disease of the upper class in Louis XIV's time. It's not. It's back, and it's horrid.
Cases of tertiary syphilis, monkeypox.
I'd be really interested to hear your thoughts on this, Martin, what you said about the cost of diagnostics and the cost of treatments on the ground. If we knew there was an outbreak ongoing, do we care? Surely we'd pay anything to stop it spreading.
You would think so, but I think what we have seen from the COVID pandemic is that everybody didn't go global. Everybody didn't treat it as a global threat. They all circled the wagons and said, okay, we're going to protect our own. And I think that was a fundamental error and actually slowed down
It didn't
the
work.
response. And it didn't work, no. I mean, by and large, most countries shut the door long after the horse had bolted.
The idea of
or in which this case, the horse came in rather than bolting
the
away.
US closed the borders to Chinese flights, and it came in from Europe. I mean, hello.
Yeah, indeed. Yeah, so there does need to be a paradigm shift, I think, in how the whole area is viewed, and in some respects, as a sort of CEO of a biotech company working at the cutting edge in discovery, you almost feel a little bit helpless because you know what you want to do, you know what you want to deliver, but finding funding for it when the market is broken is a real challenge. And what we have to do is lobby. We have to lobby governments. We have to lobby international bodies. And I think one of the things that has worried me vastly from the COVID pandemic is actually how much flack the WHO has taken in newspapers, in the public, sort of misinformation out there. It's caused major issues with credibility, which actually predicates
put
against
people at
these
risk,
things.
actually,
And
physically.
yeah, definitely.
On that note, I mean, obviously these are long haul solutions, which are you can't take your eye off the ball in terms of things like One Health, etc. But as a society, and does the political class get that, or is it always a flash in the pan? We wait till the next one. And because these major pandemics we were used to having them once in 100 years, sort of thing, Spanish flu, then COVID. We're going to see this much more frequently because of the nature of travel and intersocietal drift and all the rest of that stuff. But are we getting the message across? Do they get it, or do they?
We've had 3 pandemics in the 21st century.
Yes, exactly.
I mean,
Well, actually,
you've got to get this across. What more do you need?
It's interesting when you're talking about political pressure, because it's really something that we struggled with, because one of the companies I worked with, I was doing the infectious diseases, the other half of the company was doing Duchenne muscular dystrophy, an orphan neuromuscular condition. There's no patient advocacy, there's no pressure put on governments. There's a lot of whining biotech saying we need more money, but there aren't the patients. There isn't the National I've Had Pneumonia Foundation, is there? But if you look at the patient activism in other areas, it's hugely influential. And
that goes to the disease. Sorry, very, very briefly, the diseases. DMD is a long-term disease. Infection kills you, or it doesn't kill you, and then you get on with the rest of your life.
Or you don't have a procedure, right? Like, I think it's going to make a real impact if people realize that what we consider to be, say, routine surgical procedures cannot happen because the risk of infection is far too high. Like, if somebody is going for, I don't know, cesarean section or to get a tooth out or something that's just part of life, we know how to do it, and can't go anymore and can't have these procedures because if they get infected with a multi-drug resistant organism, the infection might kill them. I think that's when it's going to hit home big time, like in the media and for politicians, for example.
To take Richard's point, there is not a rational grouping, an advocacy group handling that. It's itty bitty bits scattered around, which have real consequences.
Because it's broad spectrum, right?
Exactly.
Like, it affects a whole load of different indications.
Until somebody famous dies.
Great. I think maybe we've talked quite a bit about sort of the current landscape and the problems we've been having in the past. And if anyone wants to raise some final comments, go for it. But then I think maybe we can start the crystal ball. And we've started to touch on that. And I think and I want your contributions here, it's what modalities. So is it classic small molecules? Do we just go into MABs? Do we use phages? Where does the microbiome come? Diagnostics? How do we link in all these really quite disparate disciplines to make sure that we have effective approaches? So I know that's a terribly open-ended question, but it would be great to sort of get your viewpoints.
If I may start, just to give you a sort of starter for 10, if you like.
Yeah, go for it.
We've got a variety of different diseases, and what we're doing is we're looking at them from a strategy point of view. And if I give the 2 I'm working on, I've got Rift at one end and I've got Nipah at the other. Nipah, we're working on a vaccine for it, be fine, kills you within 5 days. So a vaccine in an outbreak probably won't work. No. You can use it for healthcare workers, and there's obviously a need for it. Yeah. But what would you do? It's actually a combination. So you have to develop a target product profile, so such that you'll want an antiviral or some other, maybe a monoclonal or whatever. You want something that you can use in those that have got it, and then you can have prophylactic rings around it. So that's one end of the spectrum. The other end of the spectrum would be Rift, where actually a vaccine would have a massive effect. It's probably, there's a paper we published last year that actually shows of all the diseases we do, it's got the biggest impact. So depending on the target product profile, you need everything. It's not just one. And your comments are quite right about things like diagnostics. Where is the disease? Because we are misdiagnosing and we're overusing treatments, whatever it may be, whether it be an antibiotic, vaccine, whatever. We also need to know, how do you deploy it? Who's going to buy it? How much does it cost? Where are you going to build it? There's so many, but it's disease specific. And I think building those TPPs would really give you a good head start. So perhaps that might.
Well, I was thinking in terms of what you were saying about the Nipah, because that would be a very difficult business case unless you're saying, right, we know that this tends to happen here. Do we actually track the DNA of the virus in terms of those patients who had it but not died, etc.? I don't know how virulent or indeed how evil it is.
It's extremely virulent and it also mutates. And if it mutated, God help us.
So either you find a diagnostic that looks at a non-variable part of the virus, but nonetheless, the TPP is going to be very difficult, and you said it yourself, because if it mutates and it sweeps, you can't play catch up, you can't isolate, etc. So you need a therapeutic. And the question is there part of that virus which is common to other viruses that you could use a common therapeutic against? I don't know where you're going with that.
I guess I was trying to be a little bit more generalist,
Sure.
and just saying, you were also talking about bacteria and so forth. I mean, the same thing is true. And unless all of the different elements speak to each other, this isn't going to work.
Yeah, agreed.
So maybe Anne and Martin, do you want to come in here?
So, I fully agree with Mike in that we absolutely need everything. We need more of everything. But we perhaps need to be a little bit smarter as well. So, directing the right interventions to the right sort of pathogens. So, Mike was describing NEPA, that need for prevention, for prophylaxis, for the sort of ring vaccination to try and prevent an outbreak from really getting going. And so, you need things like passive prophylactics, just simple binding blockers, for instance, to give to the healthcare workers. And within Iceni Glyoscience, one of the things we're focusing on is that most viruses, particularly respiratory viruses, their first point of contact with the cell is actually the cell surface glycans, and they don't change. So the virus can mutate, but in order to infect it, it still has to bind that single glycan. So if you catenate those glycans on a polymer and stick that up your nose as a nasal spray, it can physically stop the virus from establishing in the first place. And yes, that would only last for a few hours, but for first-line healthcare workers, for instance, that could be a massive intervention. It could really nip in the bud an outbreak, to start with. So things like that, just thinking slightly out of the box and bringing all the tools to bear, I think is going to be essential as we move forward.
I think I can agree with that. So I think it's really like crystal ball gazing for the future. I think it makes a whole lot of sense to think about alternatives to traditional antimicrobials. So I can see that there could be great utility in decolonization strategies, for example, maybe rather than killing the bacteria, as you were saying earlier, or disrupting biofilm formation so that you don't get a very large bacterial load in the first place. But there's also advantages if you can just reset the ecology of that system, because maybe you're going to have a sustained colonization resistance being restored, which will give patients the ability to withstand being infected for a long time into the future, and it doesn't require any anti-infectives, like molecules, for example, that could have other detrimental effects to the existing and prevailing microbiome. Because of course, with all of the anti-infectives we have, the ones that are preserving microbiome are also really helpful because they don't disrupt the prevailing microbial communities that we're housing and holding all of the time, which means that all of the other functions that they give to us, like nutrition and molecules that help with our epithelial barriers and all the other normal functioning of our bodies, aren't disrupted either. So I think thinking of it ecologically might be a way forward. And also any interventions that would prevent like broad spectrum antimicrobials having a broad spectrum effect would be also advantageous. And I think there are a few of those being developed as well. So they'd allow the drugs to work as they were intended to work, but then mitigate the effects that are on microbiome.
So do you see a case for, take it to an extreme, if you're using a broad spectrum antibiotic and it will disrupt your gut, we know that, that the prophylactic use of poo pellets or something of that nature to effectively recolonize as soon as the antibiotic is washed.
Sure, I mean that's a thing that would be possible and would be feasible, right? To restore the depleted microbiome with a healthy microbiome again, to retrain, restore, and recreate that colonization resistance. There are also companies out there doing really interesting things, like for example, putting activated charcoal into a capsule, which would release in the colon, which is where most of your microbiome in the human body resides. So any small molecules that need to get absorbed and have effect in the small intestine can do their job. Then they adsorb to the charcoal and get washed out later without disrupting the main microbiome. So I think that's also quite inventive and pretty cool.
What about use against the endotoxins?
So there could be creative new ways to, reduce the effect or the impact of the pathogens and the drugs this way, whilst also retaining the normal functionality of the microbiome as much as possible.
Well, I think, so I'm a firm believer that we often focus too much on the MIC.
Yeah.
We're focused on what's the resistance, what's the complicated name of the bug, and how susceptible is it. Actually, the host is as equally as important as the pathogen in any infection, and microbiome, virulence factors, colonization, all of that comes into play. Again, feeds into everything we've all been saying about how we bring all of this together to go forwards in this area.
Is that this is an education thing, I suspect, the point of care element that people, as you say, is this bug going to get killed? End of story, rather than looking at it more holistically. And is that something that we need to bring into medical schools and nursing training and things like that?
Yeah, that's a panel discussion in its own right, I thought. But I know our classic approach is get the broadest spectrum antibiotic you can, give it to them ASAP, and with fingers crossed they'll still be alive in 24 hours.
Yeah.
What I think we need to get to is a much more holistic understanding of the patient outcomes, what the actual bug is, can we actually test and identify the bug at that point, and then use the right medication that's not going to have downstream consequences. But it's a big ask, it's a big step forwards, but I feel if we're going to get this area going again, these are the types of debates we need.
Perhaps there's something you said, there was a name you left out in that list. You said medics and nurses. You didn't mention vets. And the veterinary community is a bit that we are missing.
Yes, and that's where a lot of the resistance comes from.
This is where One Health comes in.
Yeah, indeed.
And this is where I think, this is the part that everybody's missing. We don't talk to each other well enough.
Animal husbandry as a whole.
Precisely. I mean, as I said earlier on, all of these things are zoonotic. We know SARS-CoV-2 is zoonotic.
Yeah.
I mean, how much more evidence do you want?
Indeed, yeah.
But the fact is the communities don't speak to each other. And then, we talk about overuse of antibiotics. Well, it's actually usually overuse in animals.
Yeah, as a feed.
That's caused.
As a growth.
But we're not speaking to each other.
No.
And until we really address what One Health actually is, because there are 3 parts to One Health, not just human and veterinary. The third part is the environment. And as the climate changes, we're going to get diseases in different places. People are living closer to animals than they should be. Guess what happens?
And mosquitoes are moving north.
Candida auris, Zika.
Precisely.
Exactly. Yeah,
You're right, mosquitoes moving north. I mean, as I say, we're about to launch another call for vaccines for Rift Valley fever, and it's co-funded by the European Union.
Right.
Why do you think that is?
Yeah, because they know what's coming.
They know it's coming.
Yeah.
Good. Okay, we've got about 10 minutes left, so I wonder perhaps we could probably take 5 minutes of questions from the audience, and then we'll do a 5-minute wrap up, and we might even finish on time due to excellent chairing. Yes. The gentleman there,
Thanks very much. It's Malcolm Barrow-Johnson from Pharma Medic. Mike, you just related to the European Commission. I remember I chaired a meeting at the Horizon 2020 some years ago, where we looked at this area completely, and actually I think we chose a GSK product at the end. And the companies went through a whole sort of beauty parade of this. It worked very well. It was a long drawn-out process for everybody at the Commission doing that. As you said, there's another round going through, another call coming through. That was a good model. Is that something we could not repeat on a wider basis? And as you're saying, we're looking at the UK, we're looking at Europe, but of course it's global, as you very rightly said. You say if you pick up something in the Yangtze's got more antibiotics than most places on Earth. It's got to be a global approach. Could it be that we put together some more of the academic units? So Horizon 2020, FP7, these are groups of academics that came together with industry to form a union, and we put together about 16 groups for that. GSK and lots of other things, including academics. Could we put together King's, Oxford, Hong Kong University, I don't know, as a global approach? It is a huge area. It comes up and up and time. We've got things like REMAP-CAP, which are trying to address these issues. So what's your view, what's the panel's issue on how we get this wider thing? I'm not saying Horizon 2020 was perfect.
No, it's a great question. I had a 2020 grant, I had an FP7, I'm that old. But it's almost like we needed, a coalition for epidemic preparedness? But anyway, that aside, I agree with you, but one of the things we've been looking at, particularly on the One Health side, is you've got to start somewhere. And actually, to say we're going to do all the diseases, you're probably not going to get anywhere. So that's why we've been particularly interested in using Rift as a paradigm, because it's actually very predictable.
Right.
If you start to detect abortions in animals, you will see human cases 2 to 3 months later. Happens every time. If we can work out what the right protocol is, and this comes back to what you guys were saying earlier on about real targeted use of it, it actually doesn't matter whether it's a vaccine, an antibiotic or whatever. If we could figure out what the protocol is, you stop the spread. And then we could apply it to other diseases. Because I totally agree with you, but if you want to do all of them in one go, I don't think you're going to get anywhere.
No, you're right, you are right.
Should we take another question? I just want to make sure we get a chance for a few
Of
people.
course.
Yes. Mike,
Oh dear.
well, this is actually for everyone, but I think your point about One Health, just to, I think that's becoming having increased focus on a national level, on government level, in the US, the UK, and that's that the health is intricately linked between animals, humans, and environment. And to mention that the Innovate UK KTN Microbiome Network is actually holding a One Health themed meeting in October. So I think that I just wanted to mention
invited.
that.
Fantastic.
Yeah.
Love to be there.
I'm happy to give you more details later. But I think that's something that has been very much overlooked in the past. I think the environmental aspect is very interesting. You mentioned obviously changing climate, but I wondered if there was any other aspects that we might be missing out on that you could comment on.
Well, this is very interesting, actually. You've just reminded me of something I came up with a rather tenuous link to this morning, but actually I think it probably fits in quite nicely here. One of the things we were doing, coming back to your question actually, if we use, say, Rift as an example, what do you need to do? You need to know where the disease is. You need to have a diagnostic. You also need to know that there are outbreaks going on. The outbreaks are linked to climate, because actually you only get Rift whenever there's a mosquito bloom after a rainy season. But because of climate change, that's changing. And one of the things we're looking at is machine learning and artificial intelligence. Because actually, what about if you were to monitor weather patterns, monitoring diagnostic data, and monitoring things like Twitter? My granny's not very well. Funny enough, my uncle's not very well. And then you start to see a pattern, and then you know where it is. And so I think all of them are linked. So you can't look at one in isolation, they're all interlinked. But I'm sorry, I'm hogging this, so please.
When you're saying about mosquito blooms, immediately makes me think of the Oxford group who put in genetically engineered males to stop the bloom from producing more females. And that fits, because if you're going to take a multimodal approach to the disease and an outbreak, we have to look at all of the parameters within that outbreak and address all of them at the same time, and that could include something like that.
Yeah, an interesting anecdote, I don't know if any of you saw this, was that recently some academics, as a joke, started looking at COVID and the loss of sense of taste and smell and complaints about food from supermarkets. And it started off as a joke, and actually they found it did correlate. So
COVID,
as people were getting infected subclinically, they were losing their sense of taste and smell. And actually you could map it onto the outbreak. And this was complaints to customer helplines for supermarkets. So that need to actually monitor what's going on globally on all the channels and pull that evidence together.
The information flow needs to come back from supermarkets, from the actual manufacturers. They're seeing peaks in this activity when they know there's nothing wrong with the product.
But this is machine learning. Exactly. That's exactly what we were talking about this morning.
Bring in the artificial intelligences. Yes,
absolutely.
Yeah.
I think we've probably got time to take one more. I'll let the microphone people decide. You're closest. Then we'll just do a quick wrap up. Go ahead.
So Michel de Baar's not on the panel, but that's not a pop at you, Paul. Because my question was, we talked about fixing the market in part of the discussion, and there was recognition that some of the things that the British government's led on with this pilot of actually rewarding innovators who would otherwise not see any incentive to move along. But the key piece in that is whether Big Pharma feels that's a sufficiently big incentive. And I just wondered, does the panel have a view on that?
You're looking at me. I think it's complicated. Push mechanisms that we have are very, very good. We're getting we can access CARB-X and Wellcome Trust and lots of other money to do research in early clinical. BARD will help you do the late clinical. Fixing the commercial model purely through pull, I think, is going to be very challenging. I like the idea of pull in principle, but the issue you have is it needs to be a global approach. You need to have consistency across governments. I'm not decrying the world order at the moment, but it's not exactly a coherent sort of tie-up. How do you ensure longevity? Who actually gets it? How we set these TPPs is quite difficult. But I do think if they're going to be developing, large pharma are going to be developing high-cost anti-cancer drugs and immunosuppressants and putting patients at risk of multi-drug infections, there's almost a pay-to-play type thing where they have to also be looking to do drug development in antifungals, antivirals, whatever. Yes, I don't know what the answer is, and if we knew the answer, we wouldn't be having this panel discussion, would we?
But effectively, you're saying to the big pharma, you have to look at your overall outcomes.
Yeah.
And if your drug causes problems, you can't walk away from that.
Yeah. So that's one possible approach, but also something you keep raising, and I feel quite strongly about, is diseases in low to middle income countries as well. We've got a real problem in the West with resistant bacteria, resistant fungi, but it's nothing compared to what's happening in the rest of the world. I think there's a long way to go, but if we get the right groups together and the right thoughts.
I mean, we were on the right track with COVAX. We didn't get it all right. We absolutely didn't, but we got close. And, that's a start. Yeah. It's not the whole solution, but at least it's a start. And the other thing that I think might be helpful is the way in which it's funded. When we fund, we don't take intellectual property from the company. We say, that's yours. So if you develop a platform and you want to use it for something else, good luck to you. We just say, we want it in the event of an emergency, you will give it to us. But if you want to use it for something else, why not? And then it's a win-win. Most of us come from a big pharma background. So we get it. You can't be too pious about this, it doesn't work.
We're basically at time, but maybe we could take just 1 or 2 minutes just to quickly go around the table. Do you have a take-home message? Either it may be what you said at the beginning, or maybe something that's come out from today, but just a take-home message for the audience.
I'll be very brief. It's complicated.
And I'll be equally brief because you're hearing far too much from me. Get everybody talking to each other. All the different parties need to talk.
I guess I'm going to say we should respect the microbiome, preserve the microbiome, and look for creative new ways to find antimicrobials.
Yeah, and I would echo that need for creativity. The point that I raised at the start, which we haven't had time to address, is where are the new interventions coming from? Where's the innovation coming from? And I'd throw out, we need some really new creative solutions, and something that has occurred to me in the past, which will probably almost never happen, is, in the States, companies can apply for R01 grants from the NIH. Proper research grants, fully funded. Why can't we have those sorts of structures in place in this country for areas where there is massive societal need, but not so much of a commercial pull? Proper grant funding for real innovation, drive things into the pipeline that the commercial world can then pick up.
Which goes back to the point is, do the politicians and society believe that is important enough?
Indeed. Yeah.
Okay, I think, hopefully you all enjoyed that. Hopefully you found it informative. Thank you, my fellow good, bad and buglies. We'll hopefully do good, bad and pretty or something next year. I don't know. But yeah, thank you very much.